Okay, we started with an amino acid. We'll have an alpha carbon. Every single amino acid must have alpha carbon, alpha carbon, alpha carbon. Every amino acid will have 3 out of the 4 terminals identical to every other amino acid in nature. It will have hydrogen terminal, hydrogen terminal, hydrogen terminal, hydrogen terminal. It will have an amino terminal, an amino terminal, an amino terminal. It will also have a carboxylic terminal, carboxylic terminal, and a carboxylic terminal. Lastly, the 4th terminal, the R group, that is the group that gives your amino acid its identity. That group is, if an amino acid is polar, if an amino acid is non-polar, if an amino acid is polar acidic, if an amino acid is polar basic, if an amino acid is acidic means charge what? Minus. Because it donated the oxygen, is going to get a negative charge. If the amino acid is basic, it has a plus charge. It is this R group, that's the group that gives the amino acid its identity. And the protein is a collection of amino acids. And these amino acids collected together with a peptide bond. So this is known as the primary first degree sequence. It is linear because it's all peptide bonds, connected peptide bonds. Then these peptide bonds, this linear sequence, is going to undergo hydrogen bonding. Hydrogen bonding in the sense that the oxygen is going, who's electronegative, is going to bond with the hydrogen who's electropositive with the other amino acid. It's not going to bind with itself. Don't think that this electronegative oxygen is going to bind with the hydrogen in the same amino acid. No, it has to be the other amino acid. And that's how they're going to fold upon each other. So extensive hydrogen bonding is going to lead to 2 types of structures. Secondary, these are alpha helix and beta pleated sheets. What are perfect examples of alpha helix and beta pleated sheets? These are your collagen and your keratin. The collagen and keratin. Then, why do we have them? Because we have a primary sequence that underwent hydrogen bonding, extensive hydrogen bonding. Then, additional extensive hydrogen bonding that will take place between the beta pleated sheets and the alpha helix when they bond upon themselves some more using more extensive hydrogen bonding, more extensive hydrophobic effects, and disulfide bridges. We will talk more about those, but these are additional bonding that carry us from being secondary structure into tertiary structure. The tertiary structure now, a perfect example of that is myoglobin. Myoglobin can now have all of these bondings in addition to ionic bonding, and this ionic bonding is going to give us quaternary, 4th degree, quaternary structure. Quaternary structure. Who is the perfect example of a quaternary structure? Protein? Hemoglobin. Excellent. Hemoglobin. It has 4 subunits. Now this is really, really important. If you denature a protein, what does denature mean? Denature means you remove, all these are weak bond. Hydrogen bond, weak bond. Hydrophobic effect, weak bond. Disulfide bridge, weak bond. We can remove them with mercaptoethanol. So if you use some form of detergent or some form of chemical agent known as you don't have to worry about that, but just remember that mercaptoethanol, if you add it, it will destroy the disulfide bridges. If you increase temperature, it will destroy hydrogen bonding. If you change the pH, if you mess up with the pH, if you mess up with temperature, if you mess up with the salt, if you add some detergent, chemical detergent, if you add some form of, if you add urea, all of these weak bonds are gonna get denatured. So now you're gonna return to primary. Can the peptide bond be denatured? it will never be denatured by these guys. So when we say denatured, it means the hydrogen bonding is gone, disulfide bridges are gone, hydrophobic effects are gone, and we return back to the primary sequence. Temperature cannot break down peptide bond unless it's really, really hot. Then you destroy the protein altogether. But we're talking about the folding. Folding is not dependent on strong bond. It's dependent on weak bond. This is so important for you to appreciate that we fold from primary to secondary to tertiary to quaternary based on weak bonding, like ionic bonding and hydrophobic effects and hydrogen bonding and disulfide bridges. But if we add some chemicals and we change the pH and change the salt, the ionic bonding is going to get destroyed, hydrogen bonding temperature is going to destroy, pH is going to destroy all of these bonds, but you're going to roll right back into your linear long hot dog, your linear long train that is connected. So denatured means we go from quaternary down to primary. But you can never break down, if you break down peptide, that's not denaturing, that's destruction. So there is a very intelligent scientist. His name was Christian Anfinsen. Christian Anfinsen, what he did was, he discovered that if he takes a protein that is in the quaternary structure, and take another protein who's in tertiary structure, then a protein in a secondary structure, denature them with temperature, with pH changes, with salt changes, with adding urea or mercaptoethanol, and adding all these chemical agents that destroy the bonds. What did he discover? He discovered that the protein denatured all the way back to the primary sequence. Then when he restored the optimal temperatures. So now he removed all the denaturing agents. He corrected the pH to give an optimal pH. He corrected the temperature, so now we have a healthy temperature. What did he find? The protein folded right back. It folded right back from primary to secondary to tertiary to quaternary. So he discovered that the most important form of the protein is not the quaternary, it is the primary. That is the form of the protein structure, that is the manuscript. That is the manuscript that is very important for the protein. That is the structure that has all the information that dictates how the protein should fold. How does hemoglobin become quaternary? It is the primary sequence. That sequence, that unique sequence of the amino acids, that is the sequence that is going to dictate how we're going to fold. So remember that it is the protein sequence that is primary. That one, if you remove all the denaturing agents, it's gonna roll and fold right back up to what it was. You add denaturing agent, it's gonna fall right back to it. But you destroy the peptide bond, it will never fold back again, because these are part of your primary structure. So these proteins give us these enzymes. So these enzymes, if something wrong with the protein enzymes inside the cell, like remember we talked about in the cell cycle, there's a protein known as P53 and protein known as RB. These are protein tumor suppressors. They stop the cell from dividing. You know, we talked about the cell cycle. We said that the cell cycle is primarily two big phases that is divided into multiple phases. So the two big phases are, first of all, you have interphase, and you have mitosis. These are the two big phases. They are divided into subphases. Interphase is divided into the G1 phase, then the S phase, and lastly the G2 phase. G1 phase is when we double the cytoplasm, double the proteins, prepare the cell for the S phase. S phase is when we double the DNA. When the DNA is multiplied by 2. The DNA is multiplied by 2 in the S phase. The G1 phase is when the structure of the protein, and the cytoplasm like get ready to be doubled. G2 is when we ensure that everything is ready to enter mitosis. Then when you enter mitosis is divided into prophase, metaphase, anaphase, telophase, and cytokinesis. So that's the cell cycle. After the cell divides, if the tissue is terminally differentiated, the cell will enter the G0 phase, known as the quiescence phase. It will not divide anymore. That's like your nervous tissue. Your nervous tissue is terminally differentiated. It can never again divide. Okay? Then you also have cells that will re-enter the cell cycle again under the condition that they need to divide. Like healing, when you're injured and the tissue needs to heal, this healing requires cell division. This healing requires that the cells divide. And also for growth and development. If a baby is an infant, going to become a toddler, going to become an adolescent, then its tissues have to enter the cell cycle. So this is very important because the cell has to enter the cell cycle. But after we heal and after we grow up, we have to stop dividing. We cannot divide forever. Who stops us from dividing is these guys, P53 and RB. These are the tumor suppressors. They suppress, they stop the cell. Tell the cell, they apply the brake. They apply the brake, stop the cell from dividing. So why do we have cancer? It's because there's a mutation in these tumor suppressor proteins, P53 and RB. They cannot stop the cell cycle. Hey, tumor suppressor, P53, hey, RB, stop the cell division cycle. We have so much uncontrollable cell division. They'll say, oh, we mutated. We underwent a mutation. We cannot stop the cell division cycle. That's why you're going to have uncontrollable growth, and you're going to have tissue that will invade these uncontrollable cell division. You'll have cancer cells that will invade nearby tissue, destroy the baseline membrane, and actually travel and enter through the blood and travel through the blood away from the bone to the lung and have lung cancer, and that is known as metastasis, to move to a far distance. But the issue is, it is those protein enzymes that destroy the cell. Even though they are below the cell, they're not part of the cell, but they destroyed the cell because they're not working. They're not doing their job. So life does not happen just at the level of the cell. It is below the cell. We also mentioned about the structural, these are functional proteins. We have structural proteins. If you have on a cell membrane, if you have a protein channel, if that protein channel is damaged, then you cannot allow the entry of glucose. That, glucose needs a membrane channel. Glucose needs a membrane channel to allow glucose to enter, known as GLUT. GLUTs are glucose transporter cells. These glucose transporters channels, if there is a defect in the synthesis and the making of these glucose channels, you will not have glucose enter the cell. The cell cannot make ATP, it will die. There are electrolytes. There are channels that allow the entry of electrolytes. But these channels are proteins. These glucose transporters are proteins. These are protein enzymes. If you cannot make them, the cell will be destroyed. You have organelles. Organelles that when they get damaged, for example, when you have misfolded proteins, when the endoplasmic reticulum is generating proteins and the Golgi body is not folding them properly, so they start piling up inside the cell, and that will destroy the cell. So these are things that are happening inside the cell. Atoms, like reactive oxidative species, free radicals, free radicals, free radicals, like superoxide, O minus 2. That O minus 2, that superoxide, that's an atom, but it will destroy the cell. Known as reactive oxide, it will become reactive oxidative species. It will cause the oxidation of the cell. These are not cells, these are below the cell. So now we're moving up from below the cell to now learning about the cell. And we have mentioned in the past that if we want to learn about the cell, we have to divide the cell into 3 different categories. We have to divide the cell into 3 different categories. The 3 different categories are your cell membrane, followed by the cytoplasm, and followed by the nucleus. Followed by the cytoplasm, followed by the nucleus. So your cell structurally could be looked at the cell membrane, and the cytoplasm, and lastly the nucleus. Now let's look at the cell membrane. We said that the cell membrane has phospholipid heads. We said that a lipid is actually looks like that. 3 carbons attached to, what's the name of this structure right here? What is the name of this structure?
Glycerol.
Yeah, that's glycerol. Excellent. Okay, and what's the name of this structure right here?
Free fatty acid.
Excellent, that's free fatty acid. Free fatty acid. So what happens is they get joined together, and we remove water. So this side gives us an OH, this side gives us an H, and we do minus H2O. This gives us an OH, that gives us an H, that's minus H2O. OH and H, and that gives us minus H2O. Actually, no, that's not true. That's not what happens. This side gives us one H, and this side gives us the whole OH. So the glycerol gives us OH, and the acid, because remember acids donate a hydrogen proton. They just donate one H. They just donate one H. They keep the O, and they donate the H. They keep the O, they donate the H. So now H2O is gone, and now we have a structure that looks like that. What's the name of this structure now? Is this glycerol anymore?
No, it's triglyceride.
Yeah, it's a triglycerol. This triglycerol is going to be modified and put in our cell membrane. What's going to happen in the triglycerol? One of the heads, we're going to remove all of this tail and add what? What are we going to add? What are we going to add to replace one of the tails with what?
Phosphate.
Excellent. We're gonna add a phosphate. What charge does the phosphate have? What charge does the phosphate have? What charge? It has a negative charge. And there is another nitrogen containing group known as we
Chloride?
mentioned that. There's a nitrogen containing group known as
Choline?
choline. Excellent.
Choline.
So this choline, so now this here is the red head. That's the red head of the phospholipid. That is the red head of the phospholipid. That is the red head of the phospholipid, and the two tails are brown. So when I draw the plasma membrane looking like that, I know that the red corresponds to my choline and my phosphate group. My choline, my phosphate group. My choline and my phosphate group. Choline and phosphate group. And the tails are my two hydrocarbon tails. These are my carbon and hydrogen. Carbon and hydrogen. These are my tails. These are my tails. These are my tails. Okay? And these, so I have, the membrane is bilipid, is a bilipid bilayer. Okay, is a phospholipid bilayer. You see that group is the phosphate, that's the phospho head, and these are the lipid tails. These are non-polar, and that is polar. You see? Okay, are we good? So this is our phospholipid bilayer. So this phospholipid bilayer encircles the cell all around. The red part is the phosphate head, and the two tails are the hydrocarbons. So now, in order for me to examine my lipid bilayer, what happens is I have to know that I have two kinds of proteins. I have proteins that are sticking only to the outside, and proteins that are sticking only to the inside, and proteins that are in both. So I have some protein that is only on the outside. This is the outside of the cell. This is the outside. That's the extracellular layer. And that's the inside of the cell. And that is known as
The intracellular.
So what's the name of these proteins?
Peripheral.
That are either on one leaflet, either on the outside or the inside.
Peripheral proteins.
Yeah, these excellent. These are peripheral proteins. Peripheral proteins. And if we want to be more specific, this is an external peripheral protein, and that's an internal peripheral protein. What about this protein that spans both?
Bless you.
Thank you. If this protein that spans both leaflets of the plasma membrane, what kind of protein is that? This is peripheral. This is peripheral protein. What about this one that spans both layers?
Transmembrane protein.
Excellent. That is transmembrane protein. It's called trans because it traverses both layers, the upper leaflet and the lower leaflet. Okay, what's a perfect example of this transmembrane protein? The first example is the leaf channels. Leak channels, they are leaking. They are open all the time. That's why we call them leak channels, because they are open all the time. Leak channels, they are open all the time. And they allow for the influx, they allow for the influx of certain ions. So you have potassium leak channels. Okay, you can have calcium leak channels. But remember, a leak channel will not allow both. So the gate in the park is open at 5 a.m., allowing anybody to go through and in both directions. Go in and go out, allow people, allow pets, allow anybody. But actually here, these leak channels, they allow one direction movement and one ion. So you have potassium leak channels, you have calcium leak channels, but you do not have both of them happening in one channel. Each one has separate type of leak channel. Then you have channels that are not leak. These channels, they are closed actually. These channels, they are closed. And these are known as what? They are closed. And these are known as, they are not leaking, so they are closed. These are known as what? Ligand. These are known as gated channels. And the gated channels, they will not open except something opens them. And something that could open them could be, for example, a ligand. So you have ligand gated channels. These ligand gated channels, they bind a ligand. They bind the ligand. This ligand is like acetylcholine. Acetylcholine. Acetylcholine is a type of a ligand. Acetylcholine is a type of a ligand that binds the gated channels, and when it binds, it's going to open the channel and electrolytes are going to start flowing down concentration gradient. So that's a ligand gated channel. There's also voltage. There's also voltage gated channels. There's also voltage gated channels. When you change the voltage, of the membrane, they get to open. They don't need a ligand. They just need voltage change. Sometimes there's heat gated channels. There's also mechanical stretch gated channels. When they feel that stimulation, mechanical stretch, or when they feel that heat, they will open and they allow the influx. So going in meaning influx, or going out, that is known as efflux.
The first one that's a leak channel.
Yeah, that's open all the time. It doesn't need anything to open. It
But
doesn't need the ligand.
The two there is open all the time. The first two.
What do you mean the first two?
Right there.
Yeah.
Those are both leak.
These are examples. These are just examples. There's leak channel for every electrolyte, but there is potassium leak channel. That means it's open all the time, and that allows the efflux of potassium, because potassium is a lot inside, so it allows efflux of potassium. Calcium is a lot outside, so this allows influx. But what we're just appreciating is that the fact that they are just leak channels. They are open all the time. They don't need something to open them.
All right.
So that's like I said, the gate in the park. This is open 5 a.m., 6 a.m., 3 a.m. It allows anybody to go inside in the park or outside the park. These are leak. But the gated ones, they are closed. So the leak channels are open all the time. They never close. It is the gated channels that are closed all the time, and they will only open when there is something that opens the gate, like a ligand, like acetylcholine, or like voltage change, or like heat, or maybe mechanical stimulation, and that causes the opening of these channels. So when you feel stretch in your body or when you feel temperature, it is because these gated channels have been opened and they allow the passage. They allow the passage of these electrolytes. That's so going, if the direction is to go inside, that's influx. If you're going outside, that is efflux. 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 Okay, the last thing we need to know about the plasma membrane. So now we learned the structure of the plasma membrane. It is made up of two tails, two hydrophobic tails, and one hydrophilic head. Two hydrophobic tails and one hydrophilic head made up of phosphate and choline. Now, the other thing we also need to know is that if we have a hormone that is a messenger that is trying to elicit a target effect. So I need a target effect in my cell. If I need a target effect, I have two types of hormones. I have cholesterol-based hormones, known as steroid-based hormones, cholesterol-based hormones, and these hormones are lipid soluble. Let me write them in brown because that's the color of the plasma membrane. So that shows you they can dissolve in the plasma membrane. An example of these steroid hormones is aldosterone, progesterone, and estrogen, testosterone. These guys, they are welcome to go inside the cell because they are lipid soluble. They are lipid soluble, so they can cross through. They are welcome to cross through, and they can enter the nucleus where the DNA is laying out there, and they can bind to certain regions in the DNA, and they can transcribe the DNA. And when we have transcription, when we transcribe the DNA, we get mRNA. And when we translate the mRNA, the mRNA, we get proteins. And these proteins will give me my target effect. So here you see that this hormone is a first messenger. It is welcome to enter the cell because it can dissolve in the nonpolar. This is nonpolar core. This is hydrophobic core of the bilayer leaflet. So what happens now, this first messenger is welcome to go inside, and it can go all the way to the inside of the nucleus to transcribe the DNA, to give me mRNA, and that mRNA will be translated to give me proteins, and these proteins will give me my target effect. So here you see the first messenger made it all the way through. But if you have the second type, and let me do those in red because they do not dissolve in the bilayer leaflet. And this is known as catecholamines. These are catecholamines, such as epinephrine. So epinephrine is a type of hormone that is polar. It is polar. Unlike the steroid, they are non-polar. These are lipophilic because they are non-polar. but the catecholamine is polar and it is lipophobic. So it cannot enter. It will only stay outside. So how can it mediate for me the effect if it can only stay outside? It will bind to a receptor from the outside, like here. Let's say that this is epinephrine. So epinephrine will bind the outside, and when epinephrine binds the outside, that will cause the transmembrane receptor to rotate and start going zzz, making some conformational change, and that conformational change will activate an internal G protein that's a peripheral protein, an internal G protein, and the G protein is going to go and activate for me a transmembrane protein known as adenylyl cyclase, and adenylyl cyclase will convert ATP into cyclic AMP, and that will be my second messenger. And the second messenger, that will be my second messenger, and the second messenger is going to activate for me some kinase proteins, and these kinase proteins will activate for me by adding a phosphate group. So a kinase will always add a phosphate group. So these kinases will add a phosphate group to an enzyme, and when they add the phosphate group, they're gonna turn the enzyme from off into an on. They're gonna activate it, start it. And when the enzyme started, it's gonna give me my target effect. So you see here with non-polar lipophilic hormones, we were able to cut through the plasma bilayer leaflet and give the target effect. But with the catecholamines, like epinephrine, we cannot enter because we are not lipid soluble. So we have to bind on the outside to a peripheral protein, and that will cause the transmembrane protein to start getting activated, and then when it gets activated, a second messenger will mediate the effect to give me a target effect. Here, we never needed a second messenger. The first messenger did everything by itself because it can dissolve in the bilayer leaflet. It is only when we are polar, when we are catecholamine, when we cannot enter and dissolve in the bilayer leaflet, then we're going to need a second messenger who's going to mediate the effect for us. So we're going to see that some more in the slides. So that's as far as all we need to know about the cell membrane. Now moving on to the cytoplasm. Cytoplasm, students confuse cytoplasm with cytosol. So cytosol is that liquid part. It is gel, that liquid white fluid gel. That's a colloid. Colloid, we said colloid is what? It's what dissolved in water? Colloid. So a solution is really tiny particles dissolved in water, like salt and sugar. That's a solution. A suspension is really big molecules, really big particles dissolved in water, such as red blood cells dissolved in plasma. Then you have a colloid, and that is when just proteins are dissolved in water. That is a colloid. And lastly, you have an emulsion. An emulsion is when you don't have a solid now, you just have a liquid dissolving in another liquid. An example of that is yeah, the oil vinegar dressing. And in our body, an example of that would be the breast milk, colostrum, and the fat of colostrum dissolving in the milk. So the cytosol is one thing, and then you have the organelles. So there are important organelles that give the cell, you see, the organelles are below the cell, but yet they keep the cell going. You have the ribosomes. The ribosomes are very important because they translate for us the mRNA to give us our proteins. We translate the mRNA to get proteins. When we translate the mRNA to get proteins, we use the proteins as enzymes, as protein channels, and so on. But if it wasn't for the ribosome, we cannot translate our mRNA. We also have the mitochondria. The mitochondria is the powerhouse of the cell. It gives the cell ATP. The mitochondria gives the cell ATP through oxidative phosphorylation. Through oxidative phosphorylation. It also kills the cell. When the cell is so damaged and it needs to be removed and properly disposed of, then the mitochondria is going to elicit programmed cell death. Programmed cell death. That's the mitochondria. You have the lysosomes. The lysosomes, they are there to fight any infectious microbe. The lysosome. So the lysosome, it has lysosomal enzymes inside. It has reactive oxidative species inside. It has proteolytic enzymes, metalloproteinases, degradative enzymes. These are all weapons, yes.
So apoptosis, that's programmed cell death?
Yeah.
Okay.
Apoptosis. That's when the cell is damaged beyond repair. It needs to be removed, and then another cell, through the cell division cycle, we have to build new cells. Otherwise, that damaged cell, that's how we get Alzheimer's disease. Alzheimer's disease is because nerve tissue is non-regenerative. Nerve tissue is staying in the state of quiescence forever. It grows one time, it stays terminally differentiated in the G0 phase. So when it gets damaged, now the cell is damaged. It must die by apoptosis. But it dies by apoptosis, but new cells cannot be made in its place. And that is what causes the Alzheimer's disease. But in skeletal muscle epithelial tissue, if this tissue is dead, then new tissue can rise in its place. Because it's regenerative, it can enter the cell cycle. And you have the lysosomes. Lysosomes are the cops. They have so many weapons, these lysosomes inside them. They have so many weapons, and the weapons, when they engulf the bacteria, the viruses, the parasite, the fungi, they add all of these lysosomal enzymes, these reactive oxidative species, and they end up destroying all of these dangerous infectious microbes. That's for the lysosomes. There is peroxisomes. Peroxisomes, from the name peroxide. So what they do is they take really dangerous oxidative species and convert them into hydrogen peroxide, which is a little better. It's the lesser of two evils. So they prevent the accumulation of superoxide, and the free radicals that have a minus lone pair and extra lone pair electrons, and they can damage the cell. These peroxisomes will take them and will convert them into hydrogen peroxide. Then some other enzymes, known as antioxidant enzymes, they will take the hydrogen peroxide, and convert it into water and oxygen. So the peroxisome is helpful because it reduces oxidative stress and free radical accumulation. In addition to peroxisome and lysosome, we have proteasome. Proteasomes, these are the shredders of damaged proteins. So if a protein is misfolded, damaged, broken, it cannot do its job, Then the cell will tag it, will put a ubiquitin tag, on the misfolded protein, and that misfolded protein will be recognized by your proteasome. And the proteasome will say, oh, you have a ubiquitin tag on you. You must be shredded. And it will shred it and relieve the cell from it. Otherwise, these damaged proteins, if they accumulate and they don't get shredded, then that will lead to like mad cow disease and Creutzfeldt disease. So cocaine addiction, substance abuse, these patients will have a lot of damaged proteins, more than the proteasome can handle. So the cell will say, proteasome, we have so many damaged proteins, come shred them. The proteasome will say, I'm already occupied. I'm working at my highest capacity. It's like when you have one cashier and 20 people waiting in line. So now you need 3, 4 more cashiers. You have reached the maximum. So now the same thing, the proteasome will say, I have reached the maximum, but there's a very severe increase in the rate of protein misfolding caused by cocaine addiction. That is the cytoplasm. And then inside the nucleus is where we will have the replication, where we will have the transcription of the DNA, and that is where we will have the preparation for the cell division. But the third major part of the cell, we did the cell membrane and then the cytoplasm. Now it's time to go over the genetic expression. Now it's time to go over what happens inside the nucleus. You have to understand that the plasma membrane is a membrane. All the organelles we talked about, the Golgi body is important for packaging, and like a vesicle, it's like pita bread stacked on each other, but they receive particles, they package them, and they ship them outside the cell if the cell is sending something to the outside. And the nucleus, and all of these organelles have membrane. They are membrane-bound organelles. The lysosome has a membrane. Inside the membrane, it has all of these hydrolytic enzymes to destroy and engulf the dangerous bacteria and microbes and destroy them. So if we now look at the nucleus looks like this. It has a membrane known as the nuclear envelope. Inside the nuclear envelope, you have your DNA. You have your chromosomes. What happens is, we know that in biology, we have DNA, and the DNA is nucleic acid, and we transcribe DNA to get mRNA through a process called transcription, and we get mRNA, and that is also a nucleic acid, and this is happening inside the nucleus. And the mRNA is going to be translated into proteins that are structural and functional. And this is translation. And this is the cytoplasm. This is happening in the cytoplasm. Okay, so if I want keratin, if I want collagen, if I want hemoglobin, if I want a protein channel, I have to go down this road. I have to go here, transcribe a region that will give me a protein channel, transcribe a region that will give me glucose transporter, transcribe a separate region that will give me collagen, transcribe a certain region that will give me some sort of an enzyme. So you cannot get proteins unless you go down the DNA. So there was a very important experiment. And the experiment was they brought a frog skeletal muscle cell. And the frog skeletal muscle cell has this nucleus, this blue nucleus. Then they went and they brought an ovum of the frog. They brought a frog ovum. They brought a frog ovum, and that's the nucleus for the frog ovum. And what they did was they removed the nucleus of the frog muscle cell and took it outside. This is the nucleus of the frog muscle cell. Nucleus of the frog muscle cell. And this is the nucleus of the frog ovum. And they switched them. So they put inside the frog ovum the nucleus of the skeletal muscle. And they put in the skeletal muscle the nucleus of the ovum. So each cell has its own structure and function. And then they hypothesized. They said, let's now push these cells to do their job. So the cell for the ovum, when you introduce a sperm, it's supposed to get fertilized and it's supposed to give you cleavage. Then you get more cells that are dividing, more dividing cells. And the dividing cells, you have blastula, then you would have gastrulation, then you would have morula, then you would have an organism. That's what's supposed to happen when the frog cell gets fertilized. When the skeletal muscle gets stimulated, electrical stimulation, it's supposed to contract, get shorter. So they said now we switch the nucleus. What's gonna happen? So the hypothesis was things are not gonna go fine. If you try to fertilize the frog ovum, it's not gonna divide. Fertilization will not happen because we don't have our nucleus. This is green nucleus. It's not green nucleus anymore. The green nucleus is over here now. And now we have blue nucleus. So things are not gonna work out. And they said, this is skeletal muscle cell. We removed the blue skeletal muscle cell nucleus. We have green, we have ovum nucleus. And when we stimulate it, it's not gonna work. Guess what happened? When they fertilized this egg, everything went perfectly. So the cell got fertilized and the cleavage happened and gastrulation happened, morula, and an organism developed. And here, when they applied electrical stimulation, it contracted. So they said, how did that happen? Then they realized that the genome, the DNA, is the same everywhere. It's like you have $1,000. You give $1,000 to a police officer, you give $1,000 to an athlete, you give $1,000 to a chef, you give $1,000 to a makeup artist. The $1,000 is the same. But if you have a question, please wait, I will tell you. Don't ask while I'm explaining. Please pay attention. If I give $1,000 to an athlete, I give $1,000 to a chef, I give $1,000 to a police officer, I give $1,000 to a makeup artist, It's the same $1,000, but are they gonna spend it the same way? The makeup artist is gonna go buy makeup supplies. The athlete is gonna go buy athletic supplies. The police officer, for example, I know they give it to them, but for example, for just this example, they're gonna go buy police supplies. But it's the same $1,000, but it is the person who spends it in a different way. Same thing, the DNA is the same. The nucleus has the same DNA. It is the cell that will transcribe different regions that will make the cell the protein it needs to do the function. So the proteins, so how does the ovum cell undergo cleavage, blastula, gastrulation, and morula and become an organism? It is the proteins that are here that makes the cell do that. And what makes the skeletal muscle cell contract? It is the proteins, not the DNA. The DNA is there, but where do we transcribe? So this part, you transcribe it, you get skeletal muscle protein. This part, you transcribe it, you get an ovum protein. So it's not the DNA. You can replace all the nucleus as long as it's the nucleus of the same organism. Obviously, if you put a nucleus of a dog cell in here, then that's different DNA. But if it's the same organism, you can replace any nucleus anywhere because it is the polymerases. These are the ones who are different. So now we have to understand that if I make a polymer of DNA, that's a DNA polymer. If I make a polymer of DNA, then who did I use? I have to use a polymerase. Polymerase. And since I made a DNA polymer, I use the DNA polymerase. I cannot use the RNA polymerase to make a DNA polymer. That's why we call it DNA polymerase, because I'm going to make a DNA polymer. And if I'm going to make a polymer of RNA, if I make a polymer of RNA, that's a RNA polymerase. And this RNA polymerase, that's an RNA polymer. Sorry, that's an RNA polymer. It is made by RNA polymerase. RNA polymerase. But you have to understand that if I'm going to make a polymer, so the polymer works here, the polymerase, I'm going to erase this ovum part. So here it is the polymerase. The polymerase is the one that is responsible for giving me mRNA. What is mRNA? mRNA is a polymer. mRNA is a polymer. So who makes mRNA for me? DNA polymerase or RNA polymerase?
DNA.
Remember, if I'm gonna make a RNA that's why they call it polymerase, because it makes a polymer of RNA. So if I'm gonna make RNA polymer, I gotta use a RNA polymerase. ACE is an enzyme. ACE. So that means I gotta use an enzyme who will make polymers of RNA. I gotta use enzymes who will make polymers of DNA. Are we clear? So if I'm going down this direction, I'm trying to make mRNA polymer. That's a polymer. mRNA is a polymer. DNA is a polymer. So I transcribe the DNA polymer to get an mRNA polymer. Who's gonna give me the mRNA polymer? Is it DNA polymerase or RNA polymerase?
RNA polymerase.
Excellent. So, but when I made the RNA polymer, who did I depend on? Who did I read to give me the mRNA polymer?
The DNA.
So yeah, that's why they call it DNA dependent DNA polymerase. DNA dependent DNA polymerase. Okay, then if we remember, I talked to you about the retrovirus. The HIV virus is going to have a reverse transcriptase. Reverse transcriptase. That's an enzyme. That's a protein enzyme. It's going to reverse and make for me DNA from mRNA. The virus does not have DNA. The virus does not have viral DNA. It just has mRNA and this enzyme. So once it infects your cell, it destroys human DNA and it causes the reverse transcription of its HIV mRNA to get HIV DNA. This process right here is mediated by what? DNA polymerase or RNA polymerase? I said, look at what are you making. Here, you're making mRNA, right? So look here, please. Here you're making, you stop writing. You see when you write, that's why you lose track. This is recorded. You can write all you want. Just pay attention, stimulate your mind. This is mRNA. I'm making mRNA. So I gotta use RNA polymerase. If I'm making RNA polymer, I use RNA polymerase. If I'm making DNA polymer, I gotta use DNA polymerase. I can never use RNA polymerase to make DNA. I can never use DNA polymerase to make RNA. So here, when I was making my mRNA polymer, what did I use? Yeah, I used DNA. I used DNA to make, I'm making an mRNA polymer. mRNA is a polymer. So what enzyme am I using?
The RNA polymerase.
Excellent. I'm using RNA polymerase to make mRNA. Who did I depend on?
The DNA.
So that's why it's called DNA dependent RNA polymerase, because it polymerizes mRNA from DNA. But this guy here, the HIV, is polymerizing what? Is it polymerizing DNA or is it polymerizing RNA?
DNA.
It's polymerizing DNA. It's going in the opposite direction. It has no DNA. The HIV has no DNA. It has just mRNA and this reverse transcriptase. So now it's gonna go this direction. So if it goes in this direction, what am I using? Am I using a DNA polymerase or an RNA polymerase?
DNA polymerase.
DNA polymerase. So you have to look at what the arrow is pointing to. If the arrow is giving me DNA, I'm polymerizing DNA, I use DNA polymer. If the arrow is pointing to mRNA, that means I am polymerizing RNA, I gotta use RNA polymerase. So this guy, HIV, is making DNA, that means it is a DNA polymer. What did it depend on?
The RNA.
It depended on RNA to give you DNA. That's known as RNA dependent DNA polymerase. That's the reverse transcriptase. So a cell, a skeletal muscle cell, okay, a nerve cell, a liver cell, is different because of the polymerase it has. So inside my nucleus of the frog, I have different regions. If the skeletal muscle polymerase is activated, it's going to transcribe this region to get skeletal muscle proteins. If the ovum polymerase is transcribed, is activated, it's going to transcribe this region. That's for the ovum. If the hepatocyte polymerase is activated, hepatocyte is going to transcribe